Good afternoon, everybody. We're going to start. It's 12:01. I'm Anna Plotkin. I'm Director of the Continuous Professional Development at the University of Toronto. Due to the large number of participants, I'm kindly asking everybody to mute their microphones. And we're starting a new series of communities of interest on molecular pathology that will consist of 4 meetings every Tuesday in July and August. This is a new format, and this is specifically done to review molecular pathology for surgical pathologists, because we think at this day and age, all of us need to correlate molecular pathology results with our surgical pathology. And unfortunately, we were not equipped with sufficient knowledge to do so. So hopefully that will help you in your journey and sign out of your cases. With that, I would like to invite Dr. Elan Hahn and Yael Fisher from Mount Sinai Hospital for a short introduction. And please, the floor is yours. Thank you, everybody, for joining. And remember to mute your microphone. If you have questions, please use chat functions for your questions. Elan and Yael, thank you.
Thank you, Anna. So I'm Yael Fisher. I'm a clinical fellow in pathology in Mount Sinai. I'm now finishing my 3rd year of fellowship. Before that, I was a staff pathologist in Israel, and my practice was in molecular pathology and perinatal pathology, and these were the topics of my fellowship as well. With me organizing these meetings is Dr. Elan Hahn, which you will hear next week. And today we're going to start the introduction to this series. So I have nothing to disclose. The learning objectives for this whole series would be to help you understand the clinical molecular reports and how to integrate those results into the surgical pathology reports. I don't think that after 4 meetings, any of us will be molecular pathologists, but hopefully you'll have better understanding of the molecular pathology reports and of the process that the specimens undergo to get to these results. Today, we're going to start with the basics. So we're going to define key terms in molecular biology, and we're going to see how to write the genomic and the protein level changes in a way that is clear to everyone, and also how to read these results. So we all know the central dogma of molecular biology. We have DNA that undergoes transcription, turns into RNA, and this undergoes translation into a protein. Now, this is the general pathway, but today we know that there are a few changes to this, and some of the RNA undergoes reverse transcription into DNA as well. But today we're going to focus on the basic pathway. So what is DNA? DNA is a double-strand helix of nucleotides. Each nucleotide is composed of 3 components. One is the phosphate group, here on the 5' end of the molecule. Then we have a 5-carbon ring sugar. That is a deoxy because it's missing a hydroxyl group here on this residue, which we will see present in RNA, and that's the difference between them. And last, we have a nucleobase that changes between the different nucleotides, and that's what gives the nucleotides their names. There are 4 types of nucleotides in 2 groups. So we have 2 purines that have 2 rings in them, adenine and guanine, and 2 pyrimidines, thymine and cytosine, that have 1 ring in them. Now, these are organized in pairs, and the pairs are constant. It's always the same pair. So adenine always pairs up with thymine, and guanine or always pairs up with cytosine. It's always a purine with a pyrimidine, and they have different bonding. So this is the only type of pairs we can see in the DNA. The strands are organized opposing each other, and there are two strands. One is the 5' to 3' strand, or the sen strand. It starts from the phosphate group on the 5' of the sugar and ends with the hydroxyl group on the 3'. And the other one is the 3' to 5' strand, or the antisense strand, that starts from the hydroxyl group and ends with the phosphate. Now, genes can be encoded on both strands of DNA. So this is a common writing of the direction of reading of the DNA. So we have TP53 that is encoded on this side, and another gene called RAP53 encoded on the opposing strand of DNA, and they have an overlap here. So the TP53 gene starts before the start of RAP53, and these areas are overlapping between the two genes. Now, just to show you this form of writing to explain it, then this is the whole TP53 gene. The plain line is the introns, and each of the thickenings is the exons. When it's in the thickest width, these are the coding exons, and when it's in between, it's non-coding exons. So this is how to read this type of writing of genes, and the arrows signify the direction of reading them. Now, many times genes that overlap also have regulatory functions on each other. So in this case, RAP53 has regulatory functions on TP53. So genes are composed of exons that are the coding regions and introns that are the non-coding regions. And each gene also has a promoter that is non-coding but controls the transcription of the gene. And all of these genes are organized in long strands of DNA that compose the chromosomes. And in between genes, there are long strands of non-coding DNA. In fact, only 1 to 2% of the whole human genome is coding DNA. The rest of it is non-coding, but it does have other functions. All of the genomic material is organized into 22 pairs of autosomal chromosomes and 2 sex chromosomes, X and Y or X and X, depending on the gender. Some parts of the non-coding DNA are pseudogenes. So you may have heard that term. So what is pseudogenes? It's sequences that are very similar to genes, but they're not translated into proteins. They may undergo transcription into RNA, and then they have regulatory functions on the counterpart genes. And it's important to recognize these because sometimes, depending on the planning of the panel that we sequence, we may capture areas of pseudogenes that are non-sequencing, and they are very similar to the original gene, but they may have a few changes here and there, and we need to be careful not to capture those as variants in the actual gene. So when we're planning and also when we're interpreting results of genes that have known pseudogenes, we have to be aware of that. So the first part of the central dogma is the DNA, and now we're going to go on to the RNA. So as I said from the beginning, the RNA nucleotides are very similar to the DNA nucleotides. The sugar is different because it has a hydroxyl group. So it will be a ribose sugar and not a deoxyribose sugar like in DNA. The RNA nucleotides compose a single strand helix. They are not paired. And they also have 4 nucleobases. 3 of them are identical to the DNA, and one is different. That one is uracil. That is the counterpart of thymine. So when we talk about RNA, instead of having a TACNG, we'll have a UACNG. There are different types of RNA. We're going to focus on the messenger RNA or the mRNA that serves as blueprints for translation into proteins. But there are other types of RNA that are very prominent in the human cells. Many of those are associated with protein synthesis, like tRNAs and rRNAs. And there are other types of RNAs that have only a regulatory function. MicroRNAs, small interfering RNAs, and long non-coding RNAs. So when we do NGS testing today, we don't really look for those, but they might serve an important function as well. When DNA turns into RNA, the first step is transcription. So the DNA is transcribed into a molecule called pre-mRNA, pre-messenger RNA, and this molecule contains the whole gene. Okay, so it'll have the exons and the introns as well. In order for it to turn into a mature mRNA, it needs to undergo splicing, a process where the introns are lost and we're left only with the exons. So each of these introns will be lost and the exons will join one another and create the messenger RNA, which will then be translated into a protein. This process is called splicing, and it might change. So not all cells that express the same proteins will have the exact same isoform of the protein. This change is called alternative splicing. And what it means is that in some isoforms of the messenger RNA, different exons are left in the mature mRNA. So this is an example of the EGFR gene, and we've already seen this form of writing. So the plain lines are introns and the thickenings are exons. And all of these are different isoforms of EGFR. And as you can see in this first isoform, all of these exons are lost. Then this exon over here is not present in into other transcripts. Some transcripts may end earlier and some may start later. But they are all a result of transcription of the same gene. After we have the mature mRNA, it undergoes translation into a protein. So the tRNA or the transfer RNA brings in an amino acid that fits the codon on the DNA. So what are these codons? They are each sequence of 3 letters in the RNA corresponds with a specific amino acid. The corresponding tRNA brings in that amino acid and creates the protein. This, of course, is not a random process. Each codon consists of 3 bases, and each 3 bases correspond to a specific amino acid. To start the transcription, there's only one codon that signal starting. That is the AUG or ATG if we're looking at the DNA, and that corresponds with methionine. Okay, so that is always the start codon, and there are 3 stop codons. Each of them terminate the translation process, and the most common one in the human genome is UGA. So when we look at a sequence of RNA that is translated into DNA, how do we know where to start the translation, or what is the specific codon? So there are many options for that, and this is called a reading frame. Okay, so this again is an example of a reading frame in EGFR. The blue lines is the actual reading frame of EGFR. So for instance, we have a leucine here, and it is encoded by CTG. Okay? But if there's a shift here, and the coding will start, instead of starting with the C residue, it'll start with the T residue, the amino acid will change to tryptophan. And if it'll start with this G, then we'll have a GGC codon that encodes glycine. So we have, in fact, 3 readings frames. But remember that both strands of DNA may encode may encode the protein. So actually, for each sequence of DNA, we have 6 possible reading frames. 3 on the sense and 3 on the antisense strand. So any change in the reading frame might create a very different result protein. Now, before going on to the rest of the talk, I would like to mention epigenomics. We're not going to talk much about epigenomics, but just to mention it, epigenomics is the study of epigenetic modifications on the DNA. These are a series of different changes that don't affect the DNA sequence, but may affect the expression. The most common at least the most studied one in cancer is methylation, and today there are tests of methylation. So just to mention that also. Now, if there are any questions about this part, I'd be happy to answer. If not, I'll go on to the next part of the talk, which will be about the types of genomic variations. So there can be many types of genomic variations, and for each one of them, there are different methods that are the most appropriate to test. We're going to focus on NGS testing. So I'm going to focus on variants that are relevant for that, but we're going to mention everything. So the first one is copy number variations, which means changes in the number of copies of a DNA segment. These may be gains or amplifications or loss of the gene, and it may be in any range of size. So it may be loss of relatively short segments like exons, or parts of a gene, a whole chromosome, or segments of a whole chromosome. The copy number variation that we as pathologists know best is HER2, or HER2, which is usually amplification, but it might not be only amplification of the gene. It might be just polysomy of the whole chromosome, or it might be both polysomy and amplification of the gene on one of the copies, although we usually refer to this as amplification of the gene itself. The next type of change is a fusion, which is also a change in the chromosomal level. So what happens in a fusion is that we have two genes, okay, gene A and gene B in this example, and there are two breaks that happen in the DNA. Usually the breaks will happen in the intronic regions just because these are far larger than the exonic regions. And they can happen in any random location. So if we look at the DNA, we have two breaks one in gene A and one in gene B, and these are joined together to create a new fusion gene starting with exons from gene A and ending with exons from gene B. This gene undergoes transcription and splicing and turns into a mature messenger RNA that contains exons from both genes, starting with gene A and ending with gene B, and this would be translated into a protein that has parts of each gene. So now I would like to ask you, and I would be happy if you can answer in the chat or just think for yourselves if you don't want to answer. Which of these following methods is the best for identifying gene fusions when the partner genes are unknown? So we have immunohistochemistry, FISH, RNA sequencing, or whole exome sequencing of the DNA. So which method would you choose? I'm going to give you a few seconds to think about that. Okay, so looking at the answers, most people write or either FISH or RNA. There are some IHC. So let's think about it. So the partner genes here are unknown. So we wouldn't know which FISH to choose because FISH uses specific probes for specific genes. We wouldn't know which probes to choose in the case where we don't know what genes we're looking for. Immunohistochemistry is the same. Many fusions have immunohistochemistry that identifies them, but without knowing which fusion we're looking at, we wouldn't know what to choose. So we're left with RNA sequencing or DNA sequencing, and in this case, whole exome sequencing. So let's look at the next slides and see. So I chose an example here of a fusion between EDV6 and NTRK3. The fusion is between exon 5 of EDV6 and exon 15 of NTRK3. Now let's look at the genes. Okay, so this is the whole ETV6 gene, and this area is the intron between exons 4 and 5. Okay, now we have a scale up here telling us how long it is. So this scale is 5 kilobases. So let's say this stretch is around maybe 12 kilobases. So 12,000 base pairs that we need to sequence in order to find the breakpoint. If we look at NTRK3, okay, so this gene is encoded on the opposite strand of DNA. So that just means there was an inversion when these two genes connected. And this is the intron between exons 14 and 15. This scale is 50,000 kilobases. So between these two genes, we have around, I would say maybe 70 or 80,000 base pairs. Now, if we want to find the breakpoint in DNA, we need to sequence at least the 12,000 base pairs from here and the 80,000 base pairs from NTRK3, and of course we don't know exactly which of the exons was involved in the breakpoint, and we're not sequencing only these two genes, we're sequencing hundreds of other genes maybe. So we will multiply this by 100. So we need to sequence around 1 million base pairs just to find or even more than a few million base pairs, just to find the breakpoints. But if we're looking at the RNA, then we have a short sequence here. We have an ETV6 that is composed of 337 amino acids, and the NTRK3 transcript starts at 529 and encompasses maybe 500 more amino acids. So let's say 1,000 amino acids. So if we sequence the RNA, we need a sequence of only we need to sequence around 1,000 or 3,000 base pairs, because this is amino acids, so we have to multiply by 3. So we're looking at 3,000 base pairs as opposed to a few millions of base pairs in DNA sequencing. So the easiest way to find fusions is to sequence the RNA when we are not sure which genes we're looking at, and not the DNA. So just to refer back to my question, so whole exome sequencing will sequence only the exons. We won't sequence the introns, so we probably will not be able to find the breakpoint if it occurs. Whole genome sequencing may find fusions because we're sequencing the whole DNA, but we will need a much higher number of reads in order to cover the whole DNA, and that's why RNA sequencing is more feasible in this case. Those were the first two types of variants, copy number variation and translocations that create fusion genes. And now I'm going to focus on point mutations, which are either single or multiple nucleotide variations, big changes of 1 or 2 base pairs, or short deletions and insertions. And there are different types of variants in this group. The first one is missense variant, which a change in 1 or more base pairs results in a change of the amino acid code. So, for example, we have the UCU that encodes serine. Okay, I can find it here in this table, and a change of the C here in the center to an A makes a change in the amino acid from serine to tyrosine. So a 1-letter change affected the amino acid. Synonymous variants are similar to these, but these do not change the amino acid. So if we look at the table here, you can see that in many cases, the third nucleotide does not affect the amino acid. So serine can be encoded with UC on the RNA, and then UC, A, or G. They will all encode the same amino acid. So if there's a change in the last nucleotide here, this will not affect coding, and that is what's called a synonymous variant. Now, while it doesn't affect coding, it might have other effects. For instance, if it's close to a splice site, which I will mention in a few minutes, then the synonymous variant might be significant, even though there is no change to the amino acid. So we should know the location of the change also, and not only the change in the amino acid. If we have a change, a true missense variant that does change the amino acid, it still doesn't mean that this will be a significant or an oncogenic change. The significance depends on mainly two things: on the location of the amino acid that changed and the chemical properties of that amino acid. So if we look at the location, and this is an example of ALK, all the blue variants here are missense variants. And you can see that they are concentrated around this domain, that if we look in the legend here, this is the tyrosine kinase domain of the protein. Other changes here are these black and white or gray and white changes, and all of these are fusions of ALK. So they can be either before the tyrosine kinase domain or rather at the beginning of the protein. But in the rest of the protein, there aren't many changes that are clinically significant. The other thing that affects the significance of the variant is the chemical properties of the amino acid change. So if there's a change in the amino acid to another amino acid in the same group, for instance, a change from alanine to valine, this would probably have less consequences than a change from alanine to tyrosine that has completely different properties. So when we're not sure about the change we have, we need to look up the chemical properties as well. Another type of point mutation is a nonsense variant. And this is a point mutation that results in a stop codon as opposed to a change in the amino acid. So in this case, we have a UAC in the mRNA that changes into UAA, which is one of the stop codons. And this causes an early termination of the protein, so the protein would be much shorter. Now, again, the significance depends on the location of the change. If the truncation or termination is close to the end of the protein, this might not be significant. But if it's in an early phase of the protein and we're losing some significant functional domains, then that might have an oncogenic result. So it's not only the nature of the change, it's the location as well. And now for a little pause to make us all more comfortable, an H&E slide that we know what to do with. And this is a poorly differentiated carcinoma in the nasal cavity. Okay? And if we look at the slide, we can see that there are two areas in this tumor. One composing of most of the tumor is a poorly differentiated tumor. And there are these two groups here that might be a carcinoma in situ. They're more confined. And this was stained for P53. And these are the results. So we have the poorly differentiated area that has null expression, has lost all expression of P53, and the carcinoma in situ areas that have also a pathological expression, right? A very intense staining of all of the cells in that area. So any ideas of what's happening here? Does anyone have, I don't know, an explanation to why we're seeing these two different patterns of staining of P53? So this tumor was sequenced, not only because of the P53. So one idea is heterogeneity of the tumor. That's a good idea. Normal tissue in the background, that might be a good explanation, but both patterns of staining are pathological. One is loss of staining, and one is strong staining. So we can assume that maybe the p53 is mutated in both of these. And indeed, we saw two types of variants in p53. So the first variant we identified was a truncation variant, okay, a termination after residue 146, and this was seen in 52% of the reading sequences. And another variant was a missense variant, also a known pathogenic variant in p53, and this was seen in far lower frequency. So the explanation is probably that the poorly differentiated component had this truncation mutation in p53. There was a loss of it, and that's why we did not have staining at all, or that we had the null pattern. And the second variant was probably present in the in situ component, and this caused the strong staining pattern. Now, this wasn't the only result we had in this case. We also found a EGFR exon 20 insertion, which helped in the diagnosis of this tumor as a tumor that is suspected to be a inverted papilloma. And if you look at the allele frequencies, this was present probably in all cells, in all tumor cells, and as opposed to the p53 that was present, where each mutation was present in a different population. Now, of course, to prove this, we would have to sequence each of the components individually. There is no real clinical significance to that, so we did not do that, but I would imagine that those would be the results if we did. The last type of point mutations is deletion or insertion variants, also known as indel variants. And this refers to deletion or insertion of short sequences. And usually when we say short, we're talking about less than 50 base pairs. But in most cases, it would be shorter than that. Duplication is a type of insertion where the insertion is of a identical sequence of DNA to the adjacent sequence. Okay, so we have two copies of the same sequence, or two or more copies of the same sequence. Both deletion and insertion variants may be in frame or cause a frame shift. So if the deletion or insertion is a multiple of 3, this will result in a gain or a function or loss of function. But if it is not a multiple, if it does not divide into 3, this will cause a frame shift. The whole reading frame of the gene will change, and this will probably result in an early termination and loss of function of the gene. So just again to show what this means, we have a normal sequence here. There are 4 amino acids, and then there is a stop codon that terminates the translation. So if we have a deletion of a U here, okay, this U, what will happen is that the first 2 amino acids would be translated correctly with met and lys, and then the third codon would change, and instead of having a UUU or TTT that encodes phenylalanine, we'll have a UUG that encodes leucine, and then it continues with the wrong reading frame. So instead of having a glycine, we'll have an alanine, and instead of having a stop codon, this will go on and keep translating into a protein until another stop codon will be formed. If we have an insertion, again, in this case, an insertion right after the first methionine, the first AUG codon will have an extra U. This will again cause a frame shift because we have an insertion of only one base pair, and this will create an immediate stop codon in this case and terminate the translation right here. In other cases, if this is in the middle of the gene, a stop codon might be formed earlier than expected, and the gene would be truncated early. Now, if we have an insertion or deletion of three nucleotides or any multiplication of three, what we'll have is loss or gain of a few amino acids, but this won't affect the rest of the reading frame. So in this case, we have a loss of AAG that encodes lysine, but the phenylalanine and the glycine and the stop codon after them continue to be translated appropriately. So this might affect the protein less. Sometimes, depending on the location, this might create a gain of function for instance, the tyrosine kinase domain. That's a fairly common type of variant in those domains, and it might not have a clinical significance at all if it's not in a functional domain. Now, I mentioned splice sites before, so let's talk a bit about that. Splice sites are the areas around the exon-intron boundary. Now, this form of writing is a conservation form of writing, where as the letter is larger, it means it's more conserved across species. And in this case, we're looking at splice sites or exon-intron boundaries in the human genome, and most of them would be on the 5' end, AG on the intron side, and then GT in the exon side. And you can see that the GT is more highly conserved and is actually present in all of the splice sites. And if we look at the 3' end of the exon, we can have we always have AG in the end of the exon, and the letter before that might be CT or A. And usually in the intron side, we'll have AGT. So we have AGGT and AGGT on both sides of the exon. When we're talking about splice site variants, we're talking about usually variants that are between -14 or 14 base pairs before the start of the exon, or 6 bases, or depending on where you look at, sometimes it's 11 bases after the end of the exon. So we have -14 on the 5' side and 6 or 11 base pairs on the 3' side of the exon, and any variant within this range might affect splicing. Again, usually the affected amino acids are in ±2 around the exon, because as you can see, this is the most conserved area. Now, a good example for a splice site variant is the MET exon 14 skipping mutation. And in this case, different mutations around exon 14 of MET can cause this variant. So what we see in the DNA are the variants themselves. These might be insertions or deletions in the intron or spanning the intron and axon boundary, like in this case, or even point variations in the two base pairs around the exon and intron boundary. So all of these mutations result in the same thing, in the same consequence, which is skipping of exon 14, and I will show you how that works. So in the normal splicing of MET, the peach exon here is exon 14, and when all the splice sites are normal, the mRNA will have all the exons around exon 14, so 12, 13, 14, and 15. Each one of these has a different functional significance, and exon 14 encodes a domain of the MET protein that is called the CBL binding domain. Now, CBL is a ubiquitin ligase of MET, and when it binds to MET, it leads to lysosomal degradation of the protein. Now, when we have aberrant splicing of MET, in this case, because of mutations around the splice sites of exon 14, we'll get an mRNA sequence that does not include exon 14. This sequence will be translated into the MET protein, but this MET protein will lose the CBL domain. So we're going to be left with a MET protein, right? A tyrosine kinase that does not have a binding domain to its ubiquitin ligase, and it will not be degraded, and it will stay highly expressed on the cell membrane, causing overexpression and the oncogenic effect of MET in this case. So that might be a result of aberrant splicing. Of course, other splice-like mutations may have loss of function effects on the protein, again, depending on the location of them. So I showed you different types of mutations, but there are other terms related to mutations that we all need to know. So this would be a brief review of them. We have germline mutations and we have somatic mutations. Germline mutations can be inherited from the parents, but they might also form in the germ cell. Those would be de novo mutations, but they would be present in all cells of the body. Somatic mutations are present in a subpopulation of cells. They are formed after the fertilization process and can be present in one or more types of tissue. In our context, we always think of somatic mutations as mutations in tumor, but that's not necessarily the only type of somatic mutations we have. Different genetic disease can also be a result of somatic mutations, and it depends on when and where this mutation occurred in order to determine what will happen at the end. Usually when we talk about mutations in cancer or variants in cancer, we're thinking about driver mutations that contribute to the malignant phenotype. But actually passenger mutations are far more common. They are randomly distributed in different genes or in different locations in the DNA, and they do not have a phenotypic consequences on the cancer cells. Another term you may have heard is polymorphism, which is two or more variants of DNA sequences among individuals, in the same individual and between different individuals in the population. A SNP, what we call, is a polymorphism involving a single nucleotide. And this is a very common type of variation between human beings, where, of course, not all of our DNA is identical. There are different databases that help us to recognize these polymorphisms. One of them is NOMAD, and we'll talk more about that in in the next talks. But for instance, this is the BRCA2 gene. All of these circles or ovals here on the gene are known polymorphisms in the gene. All the green ones are synonymous, meaning they are probably benign. Okay. Others are missenses that are known as benign as well. And only a few of them cause loss of function. These are these pink peachy variants that you can see here. So if I sorted this out according to the most common variants, and you can see that all of them are benign, some of them are missense variants, and this first variant was found in more than a million and 6,000 people, and 800,000 of them were homozygotes for the variant, meaning this variant was present in both of their copies of BRCA2. So if it's present in so many people in the general population, this is probably not a clinically significant mutation, and it is indeed classified as benign, as are all the other common SNPs in BRCA2. Some of these are synonymous variants, others are changes in the intron, but they are all benign. So after we heard about all the types of variants that exist, let's look at the nomenclature or how these are written. So these are a few examples that the ones of you that answered our survey have seen. And in general, the way we write variants is we start with a gene symbol, then we have an accession number. Sometimes it's the other way around. It doesn't matter. It means the same. And we show the coding change and the protein change. So before we start reading these variants, we first need to know which genome version we're working at. So what is a genome version? It's the reference sequence of the normal DNA. And over time, versions are updated. It takes a few years, of course, to update a new version of the whole genome. The most common version that is used in most clinical laboratories is HG19 or GRCH37. This came out at 2009. In the research setup, they use GRCH38 more. This came out at 2013. And the newest version of the gene is called T2T, which is telomere 2 telomere. And that came out in 2022. It's still not in wide use. So if you get a report from a clinical laboratory, most likely you'll get it in HG19. And when you look in databases to find information about the variants you're interested in, you need to make sure you're looking at the same genome version. Okay, so back to the nomenclature. So the first few letters are the gene symbol. And these are short abbreviations of the gene name. Many times they're different from the protein name. So for instance, the gene name TP53 encodes the protein P53. It's custom to write gene names in italics. So if you're not sure whether they're talking about a gene or a protein, if it's italicized, it's probably the gene. And different genes, or some genes may have more than one symbol. So BRCA2, which is the most common name of this gene, is also FANCD1. So if you're not sure what gene you're talking about, check maybe it has a more common name that you do know. The second part is the excision number, and this is a unique identifier for a genomic sequence. There are different types of excision numbers, but the most common ones are the RefSec excisions, and that's what you'll usually see. These start with N and then a different letter, and then the number of the sequence. Usually, we'll look at NMs, which are ref seq sequences of messenger RNAs, and they will have a number and then a dot to a version. The version sometimes changes, but these changes are usually not very significant, at least not in the areas that we're interested in. So usually what you'll see is the mRNA code, but there are different codes for non-coding RNAs, for proteins, for the location on the chromosome or the genomic location. So what does this mean, the different excisions? So I showed you before that different transcripts of the same gene may contain different exons. And the transcript number or the excision number reflects that. Usually there is one number that is the consensus, and it might be identified as the main select, and that is what you'll usually use. But when you look up variants, you need to make sure you're looking up at variants that are on the same location. So, for example, this is a common mutation in exon 20 in lung adenocarcinomas, T790M or threonine 790 methionine. And in different transcripts, we're looking at the NM. So this encodes the coding spot, and you can see that it can be in different transcripts and different numbers. Okay, so we have the main select is 2369, but in a different transcript, this might be in 1568. And if we are not aware of the transcript number, we might make a mistake in interpreting this variant. And similarly, the coding number might also be different. So if usually we refer to this as 790, in another transcript, it might be 745. So this number is very important to notice. The next part of the variant is the coding number or the coding change. And this is written in this pattern. So we have C for coding, then we have the number of the base pair that we're referring to. And in a missense variant, we'll have the original nucleotide that is changed into a new nucleotide or the variant nucleotide. So for instance, if we're looking at KRAS coding variants, there are 6 common hotspots on KRAS, and these are the most commonly involved bases that are altered. So if, for instance, if we look at base number 34, and there's a G there, if we have a variant, it might be, for instance, G2T. So what we're talking about is a change of the G in position 34 into a T. Now, how do we know what 34 means? So when we refer to these numbers, we're talking about only the coding DNA. How do we find that out? So if we look at the whole protein in this form of writing, the small letters are the introns and the capital letters are the exons. The red letters in the exons are non-coding, and then the blue letters are coding. So this is exon 2 of KRAS, okay, and it starts as non-coding. And then we have an ATG code, which is the start codon that encodes methionine. And we start counting from this first A. So C34G means this G over here. If we look only at the mRNA, again, we have in red the non-coding part of the mRNA, and then we have the ATG starting the coding, and we start counting from this A residue. Each of these columns is 10 character wide, so 1, 2, 3, 30, 1, 2, 3, 4. This is the G that we're looking at that is altered. And another view of that in a genome browser, we're looking at residue 34. Over here, it is this G that is the first letter in the G12 codon, right? Because the reading direction in this case is from right to left. We have this little arrow here that shows us the reading direction. When we're talking about indels, then the nomenclature is similar. Okay, for an EGFR duplication here, we have C standing for codon, and then the two positions in between them, the duplication occurred. So we have a duplication between 2290 and 2291 with an insertion of this sequence that is identical to the sequence around this area. In the case of deletion, we're starting with the deleted with the deleted base pairs. So in this case, the range is between 1735 and 1737. We have 3 base pairs that are deleted, and these are the 3 base pairs. The G would be in position 1735, A in 36, and T in 37. And of course, we list the base pairs that were deleted after. In some cases, you will not see the base pairs, you'll see only the numbers in cases of deletion. Of course, when there's an insertion, the numbers, the letters should be present there so we can know what was inserted. The next level, is the protein change. Okay, so that's the last part of the variant name. And in a missense, this will be written as P dot and then the original amino acid, the codon number or the amino acid number in the protein, okay, and the variant amino acid. If we have a asterisk or the code TER termination, it means the variant caused a stop codon. There are 2 different ways to write the amino acids. One is a 3-letter code or a 1-letter code. Usually, the 3-letter code is preferred. It's better practice to use the 3-letter codon. But sometimes this might be confusing because for many variants we refer to them as the 1-letter codon. So we all know BRAF V600E. I'm sure all of us heard about that variant, but when you write it in a 3-letter codon, you'll have a valine 600 to glutamate, which GLU. I had an oncologist that was very experienced, but every time she saw this, she got confused and she was sure we're talking about a different variant in BRAF and not V600E. So we should be minded to that, even if we do use the 3-letter codon. In some cases, that might be confusing. So it's maybe better in these cases to use both types of writing. Again, to the variant that we saw before, so I showed you a change in KRAS 34G to T, and this changes the codon from GGT to TGT. So GGT encodes glycine or G, and the change causes it to encode cysteine or C. Okay, so the protein change would be glycine 12 to cysteine. When we're talking about insertions or deletions, then it's a bit more complicated to translate it into the protein. So in this case, we have a duplication inserting this series of base pairs into right after base pair number 2290. And sometimes it's not exactly the reading frame that we see here. So in this case, the reading frame actually started one base pair before the TC that was inserted. So the first code was TTC that encodes phenylalanine, then glutamine, glutamate, alanine, and tyrosine, which was the original. So instead of alanine and then tyrosine, we have an insertion of these four amino acids into the protein, and that is an exon 20 insertion variant in EGFR. In this case, this is a deletion in the KIT gene, and we have a deletion of GAT. GAT encodes aspartate, so we have a deletion of only this amino acid without any effect on the amino acids around it. Sometimes these insertions and deletions are more complex. We need to be minded to the reading frame. So it's important to recognize if this causes the complex insertions and deletions affect the reading frame. So this is an example of an deletion and insertion into KIT. There's a deletion of 4 base pairs and then an insertion of one of them. So in total, this is a in-frame variant because we have a change in only these 3 variants. And instead of having the original code that was TAC and then GAC, we have a deletion of these 3 ACGs and then a change of this A into C. So instead of encoding 2 bases, tyrosine and aspartate, there's a change in the coding in one codon, and it changes into only serine. Okay, so we lost one amino acid and one is changed into serine. Another term you're going to find on molecular reports is the allelic frequency or variant allele frequency. Allele is one of 2 or more versions of DNA sequence. It can be a version, a different version in one single base or in a segment of bases, but it's always at the same given genomic location. So an allele is a different copy of the DNA, essentially. When we're talking about somatic variants, we would like to know the variant allele frequency, which is the incidence of a variant in our test results. Now, germline variants will always be around 50%, okay? But somatic variants, their frequency will depend on the tumor cell content. When we have oncogenes, these are usually gain of function variants, and we expect them to be present in half of the tumor cell content because usually only one copy of the specific gene is affected. In tumor suppressor genes, we'll usually see loss of function, and that can be in any variant allele frequency. Sometimes it would be in 50% of the tumor cell content. Sometimes it'll be in all the tumor cells. When the VAF is different than what we expect, of course, we need to go back to our estimation and make sure that we estimate it correctly. But if we did, we should consider whether there are copy number variations or heterogeneity in the tumor that cause this different variant allele frequency. So tumor cells are genetically heterogeneous, and variants are not always present in all tumor cells. Usually the first mutation that drive the carcinogenesis will be present in most of the tumor cells, but tumors evolve all the time and other variants may not be present in all tumor cells. When we estimate the percentage of tumor cell content in the slide that we examine, we need to look at the tumor nuclei and not at the tumor mass. Okay, so, of course, tumor cells might be different in size than the cells surrounding them. When we have large tumor cells surrounded by many small immune cells, it might be confusing. The area is very different than the percentage of the tumor nuclei, and inside the tumor mass, we might also have fibroblasts, blood vessels, etc. So always try to look at the tumor nuclei and not at the mass, because at the end of the day, the nuclei are what contains the DNA, and that's what we're testing. Another term you must have heard about is tumor mutation burden. And what this means is the number of somatic mutations per megabase of the genomic sequences that we interrogated. It's relevant for immunotherapy, and it's ideally estimated in genes that are not associated with cancer. So we're just looking at the random variation in the tumor DNA. Of course, if we have any variants that look like they're in the germline, we should exclude these because these are not changes in the tumor. And when we have a high tumor mutation burden, this might be related to deficient repair systems like the MMR or the HRR systems that I will talk about in a minute. But the quality of the number we receive in the tumor mutation burden testing depends on the panel that we're using. So there are different panels that test for TMBs. Each of them uses a different number of gene, covers a different number of base pairs, and some include only non-synonymous variants, others include synonymous variants. So they are really different. And that's the reason why cutoffs for treatment are difficult to establish. So this is a paper that compared the different panels, and the confidence interval for TMBs really varied greatly based on the size of the panel that was used. So when we have a lot of variants, then it's pretty easy to see that the TMB is high. But when the number of variants is lower, the confidence interval varies, and you can't really trust the panel, or you don't get consistent results between the different panels. So just in a couple of words before I end, to mention the repair systems that I mentioned. So microsatellite instability, we've all heard that term, I'm sure. Microsatellites are short repeated sequences of DNA, so it might be a repeat of a single base or two doublets or triplets of bases. And microsatellite instability refers to a change in the number of in the length of those repeats, and it's a hallmark of mismatch repair deficiency. So when there is DNA replication, and the areas of repeats are prone for errors. The mismatch repair system, if it's intact, if it's proficient, it should correct those errors, and the length of the microsatellites would not change. But if this system is deficient, there will be instability in the microsatellite, and the length of these repeats would change. Okay. So we can test for microsatellite instability using IHC for the MMR proteins. We can do a PCR test for areas of known microsatellites, or we can do it on on NGS. Microsatellite testing is included in some of the commercial comprehensive NGS panels, but it doesn't always work as well as the PCR-based tests that we know. These changes might result from variants in the MMR genes. These might be hereditary variants, and we can pick up those variants if we sequence the MMR gene, or they might be a result of methylation or epigenetic silencing of these genes, specifically MLH1, and we can test for that methylation separately. The other repair system is the homologous recombination repair system that repairs DNA based on copying the homologous area. It repairs double-strand breaks in the DNA. And deficiency in this system is associated with mutations in BRCA or in the BRCA genes. When we test for homologous recombination deficiency, we can either test for the pathogenic variants in the BRCA or BRCA genes, or we can test for the results. These tests are quite complex, and they include three components: loss of heterozygosity, telomeric allelic balance, and large-scale transitions. This requires more complex testing than the usual NGS panels, and that's the reason not all NGS panels cover this genomic instability testing. So we're almost out of time. What we talked about today was an introduction to molecular pathology. We talked about the central dogma of molecular biology. We talked about types of variants in the DNA, about the nomenclature, and a little bit about genomic instability. So I hope this was useful. I want to thank you for your attention, and if there are any questions, I'd be happy to answer.
Thank you so much, Yael. I can see a lot of great comments, and thank you for your very informative webinar. It's honestly one of the most informative webinar I've ever seen. So really much appreciated. I think it's a great baseline for everybody to follow in the following 3 meetings that we're going to have starting from next Tuesday, another 3 Tuesdays. People to remember to log in for the consequencing for the entire series. And also, there are a lot of questions about recording. We will be posting all the recordings on the University of Toronto Laboratory Medicine Program CPD website. It's free and everybody can have an access. With that, I think Elan and Yael, I'll let you answer any questions that you can potentially have. I don't see it, but maybe Elan, I know you answered a lot of the questions in the chat while Yael was presenting.
If there are no questions, if there are questions in the next talks, I'd be happy to answer them then, if anything comes up.
Sure. You have 1 week to collate all your questions. Okay, I don't see anything. Thank you very much, Dr. Fisher and Dr. Hahn, for this comprehensive start. We're looking forward to seeing you in a week. And with that, we'll finish the webinar. Thank you, everybody.